In our quest to decode the genome, we employ a powerful arsenal of tools and techniques. Intrinsic gene annotation is one such approach, relying solely on the information encoded within the DNA sequence itself. It's like deciphering an ancient language using only the patterns and structures within the text. Intrinsic methods operate under the premise that genes leave behind telltale signs of their presence. These signs, embedded within the DNA sequence, serve as beacons for gene prediction algorithms. One such sign is the presence of open reading frames, or ORFs. ORFs are stretches of DNA that have the potential to code for proteins. Another crucial clue lies in the distribution of nucleotides, the building blocks of DNA. Genes exhibit characteristic patterns of nucleotide usage, distinct from non-coding regions. Intrinsic methods exploit these patterns to distinguish between the two. They employ sophisticated statistical models to identify regions with a high probability of being genes. However, intrinsic gene annotation is not without its challenges. Genes can be short, their signals faint, making them difficult to detect. Moreover, the rules governing gene structure can vary significantly between organisms. This variability adds another layer of complexity to an already intricate puzzle. Imagine trying to understand a foreign language without a dictionary or any prior knowledge. That's the challenge faced by AB Initio gene prediction methods. These methods rely solely on the inherent properties of the DNA sequence to predict gene locations. They are the true pioneers of gene annotation, venturing into the uncharted territories of the genome. Ab initio methods employ a variety of algorithms, each with its own strengths and weaknesses. Hidden Markov models, or HMMs, are a popular choice. HMMs are statistical models that excel at identifying patterns in sequential data. They learn the characteristic features of genes and their surrounding sequences, using this knowledge to predict gene locations. Another powerful technique is neural networks. Inspired by the structure of the human brain, neural networks excel at pattern recognition. They can be trained on vast data sets of annotated genomes, learning to distinguish between genes and non-coding regions with remarkable accuracy. These networks are constantly evolving, becoming more sophisticated with each passing year. Despite their power, ab initio methods face significant hurdles. Genes can be short and their signals subtle, making them difficult to detect. Moreover, the rules governing gene structure can vary significantly between organisms, adding another layer of complexity to the task.